Thank you very much, and thank you very much for the invitation to be here. It's great to be here, and so I've been asked to speak on various types of IVM systems, including this IVM system called SPOM. So this is, I'm really happy to give this presentation because there's been so much progress made in animal work over the last 10 years or so on developing IVM systems, and there are now many different types of IVM systems, and I'm going to speak about some of those, but I'm also going to talk about the challenges, the really very substantial challenges of taking this into the human clinic, and why it is that we haven't really made a lot of progress in terms of getting it into the clinic, and hopefully we should be able to change some of that, and this is the right corner of the world to do that, I think, to make some progress on clinical trials in human IVM. So, I just want some very simple basics to start with. I just want to remind you that Marc-André Serrat has introduced this very nicely for me, but just want to remind you that IVM differs from IVF because the oocytes are collected at the immature germinal vesicle stage, and they're matured in vitro as an intact cumulus oocyte complex for 24 to 40 hours to generate a metaphase 2 oocyte, which is then treated in terms of from an ART perspective exactly as you would for IVF. So maturation occurs in vitro, and secondly, the oocytes are collected typically from smaller follicles. So that's the essential difference between IVM and IVF. So where are we at the moment? So roughly there are about 7 million human IVF offspring that have been produced from IVF. We don't know how many human offspring have been produced from IVM, but we estimate that it's about between 5 to 10,000. So obviously IVM plays a very small role in human ART compared to IVF, and that's curious and challenging because you can see in the domestic breeding sector, IVM is the reproductive technology of choice over IVF. And you can see here that in cattle breeding, at least 400,000 embryo transfers occur with IVM blastocysts in cattle per year. So there are literally millions and millions of IVM cattle produced from IVM when there's almost none produced by IVF. So it's very interesting that you have this dichotomy that IVM is the preferred technology in domestic species, whereas IVF is the preferred technology in humans. So why is that? That's really, really very strange. So here are some of the latest results from the cumulative live birth rate per oocyte pickup in human. And this is from Australia, from Perth, from Roger Hart's group. And this is an exceptionally good IVM unit, and you can see here that the cumulative live birth rate is around 40% from IVM. So these rates are very good, I would say, very respectable. And really there's only a gap here of about 15% between IVM and IVF success rates in PCOS patients here. And I'm, of course, aware that clinicians, patients, and the clinics are exquisitely sensitive to success rates in human ART. And I think this difference of just 15% between IVM and IVF is the principal reason why IVM is not used. The other thing I would say here is that the IVM system that's used in Perth, in Australia, is a very good one, but it uses human serum in the IVM phase. And that's obviously not something that could be used or wouldn't be expected to be used worldwide. We really need to move away from the use of serum in human IVM. But nonetheless, this is probably as good a success rate from IVM as you see anywhere. I should emphasize this is single embryo transfer that's used for IVM here. So, the human literature is greatly complicated by understanding what IVM is. This is not complicated in the animal literature. There is a standard definition of IVM, which in fact was set up by Bob Edwards more than 50 years ago, and that is that human oocytes are matured in vitro, as the name suggests. Okay, so they are collected at the germinal vesicle stage, and very importantly, they're cultured as intact cumulus oocyte complexes. Okay, so this means that we have an intact cumulus oocyte complex at the GV stage at the start of maturation, and then it progresses through metaphase 1 to metaphase 2. So actually IVM is not so much about the oocyte, IVM is about the culture of the cumulus cells and the interaction between the cumulus cells and the oocyte. So IVM of denuded oocytes is nonsense. So this idea that you can collect denuded oocytes from an IVF cycle and culture them is not IVM at all. These oocytes are certainly not compatible with pregnancy. So consistent with the standard definition is that FSH priming is compatible with this because, of course, as Marc-André Seurat has just showed you, FSH priming has very important roles in the development of the oocyte in vivo, which is consistent with the original definition. The human literature is greatly confounded by the use of HCG priming, whereby this, of course, induces oocyte maturation in vivo, and a substantial body of the literature uses this system, which has caused a huge amount of confusion in the human ART sector. And I should emphasise that this is never used in any of the animal studies. So I'm going to be talking about the standard understanding of IVM for the rest of this presentation. So there are many reasons why IVM has proved challenging, and I'm going to talk mostly about the biological challenges today. And I see them as really two fundamental biological challenges. And one is that oocytes are typically collected from small antral follicles which are not fully developmentally competent. And this is exactly the topic that Marc-André spoke about previously. Unfortunately, I'm not going to talk about that too much today. But also that maturation in vitro occurs spontaneously necessarily without appropriate somatic and maternal control. The point being that oocytes are collected from small follicles and undergo a type of artificial oocyte maturation. So I'm just going to have 2 slides on this first point, and the rest of the presentation will be on this second point. So Marc-André has presented this beautifully. Follicle size is really, really important for the overall acquisition of oocyte developmental competence. Oocytes from small follicles, which are typically the oocytes that we use for IVM, whether in domestic species or in human IVM, have relatively low developmental competence for the reasons that we've heard about. Whereas as follicles progress through folliculogenesis, they progressively acquire the capacity to support embryo development. There are really, really important molecular changes that occur within the oocyte. In vivo, this is driven particularly by FSH and associated cyclic AMP pathways. We know now many details about how the oocyte maturation molecular cascade is regulated. We know that oocytes from small follicles are transcriptionally active. They're still acquiring the machinery that they need to support embryo development. And very importantly, these oocytes from small antral follicles are EGF-like peptide unresponsive. So the LH surge that induces oocyte maturation, the essential molecular cascade that's responsible for or is associated with ovulation and oocyte maturation, is the EGF-like peptide signaling cascade. And oocytes from small antral follicles, even though they're fully grown and fully meiotically competent, are EGF-like peptide unresponsive. And as we progress through folliculogenesis, those cumulus oocyte complexes become EGF-like peptide responsive, and we know now that that's through a combination of key signaling systems downstream of FSH, so particularly through cyclic AMP and PKA signaling, but also very important signals, paracrine signals coming from the oocyte in the form of GDF9 and BMP15, which form a nexus at the EGF receptor that plays a very important role in the acquisition of developmental competence. So now just to return to this concept of spontaneous maturation. So a really fundamental point to note is that when you collect an oocyte from a small antral or even a large antral follicle, that oocyte, if you take it out of the follicle, put it in vitro as a cumulus oocyte complex, it will typically spontaneously meiotically mature. That is, you simply put it in culture, particularly in the case of mouse, for example, it will go through to metaphase 2. That doesn't mean that it's a good quality oocyte. So the typical sort of culture systems that we use are complex culture media, that is, they're tissue culture media. We usually have some type of protein supplement, can be serum, or can be albumin. FSH plus or minus EGF or EGF-like peptides. So this is a really very, very simple system, and unfortunately most of the clinical IVM that's still done around the world is using this very traditional, very standard system that doesn't really support the appropriate levels of oocyte maturation that we require. So some basic principles of the work from my lab and from many other labs from around the world. is this concept of oocyte capacitation in vitro. So this is actually trying to replicate many of the processes that Marc-André Serrade talked about that occurs in vivo under the influence of FSH, to try to replicate that in vitro. And a really fundamental principle here is that for good quality IVM systems, we need to prevent spontaneous resumption of meiosis as soon as the COC is taken out of the follicle. So the oocyte will normally undergo germinal vesicle breakdown very quickly, and that needs to be prevented. Okay, and the reason why it needs to be prevented principally is to maintain cumulocell oocyte gap junctional communication. This is associated with an ordered cessation of RNA synthesis, as we've heard previously. So this is really important for the oocyte to be in a state in which it can undergo maturation and development properly. So this is actually relatively easy to do, and there are many advanced systems in which this can be done. So firstly, we need to use a meiotic inhibitor, and there are many different types of meiotic inhibitors. I'll speak about that. We also need to support the cumulocells during this period. Now that's a question that's open for discussion, but certainly a little bit of FSH is good. Some of the oocyte secreted factors are very valuable, and of course the basic composition of the media is really important. And this means that this becomes a bit of a cocktail, and this is one of the challenges, is that there are many interacting effects here. So let's talk about some of the meiotic inhibitors. This work has been around for a long time, but we now know really some very great details of how oocyte maturation and how oocyte meiotic resumption is regulated. So firstly, we know that the oocyte is maintained under meiotic arrest because of high levels of intra-oocyte cyclic AMP. We know that the oocyte possesses a very potent phosphodiesterase, and that phosphodiesterase needs to be kept under control. So we can use various phosphodiesterase inhibitors to keep the oocyte at the GV stage, or we can use exogenous cyclic AMP, so that high levels of cyclic AMP in the oocyte come basically from two sources. One is the G protein coupled receptor, so endogenous production of cyclic AMP inside the oocyte, and also, of course, the cumulocells have the capacity to supply the oocyte with huge levels of cyclic AMP through gap junctions. Very important breakthrough a couple of years ago through some very significant mouse papers published in papers like Science and Nature, not from my lab, I should emphasise. So we know now that cyclic GMP, which is supplied by the cumulocells, is the principal molecule that inhibits the activity of the phosphodiesterase. So cyclic GMP is a key molecule inhibiting meiotic resumption. And cyclic GMP is produced under the influence of C-type natriuretic peptide. So John Eppig discovered this together with some colleagues in Beijing. And this is a really important concept because we now know how oocyte maturation is maintained. How oocyte arrest is maintained. So this is what occurs during meiotic resumption in vivo. This is fairly complex, but I just want to illustrate a very simple point. Of course, oocyte maturation is induced by the LH surge. We now know all the molecular, or a substantial amount of the molecular details of how the LH surge is transmitted through to the oocyte to cause the induction of oocyte maturation. So particularly through the action of a fall in C-type natriuretic peptide, we get a reduction in cyclic GMP from the somatic cell compartment, and then the oocyte phosphodiesterase is activated. The second point I want to illustrate here is that you can see here that clearly in response to the LH surge, there's a gigantic molecular cascade that's induced in the maternal compartment of the follicle in the somatic cells, which are transmitted to the oocyte. So there's all sorts of really important molecular signals that occur here during oocyte maturation which don't occur in vitro. So because we now know what happens in vivo, we should be able to replicate some of those in vitro. So one of the very interesting anomalies here is that even though I said that a drop in cyclic AMP is required for oocyte maturation, in the whole cumulus oocyte complex, oocyte maturation in vivo is associated with a very large increase in cyclic AMP because the second messenger for LH is cyclic AMP. So normally oocyte maturation is associated with an increase in cyclic AMP followed by a decrease. But under standard maturation IVM systems, we have a drop in cyclic AMP, and that's why maturation occurs. This small increase here is associated with with the addition of FSH. So using standard IVM systems, we have a drop in cyclic AMP, whereas in oocyte maturation in vivo, we have large levels of cyclic AMP. So why can't we replicate that in vitro? And in fact, we can quite easily. So one way of doing that is to use a phosphodiesterase inhibitor during the IVM phase. So this is an induced IVM system whereby the oocyte is in fact arrested using a phosphodiesterase, such as, for example, milrinone in the IVM phase, and oocyte maturation is induced in vivo using something like FSH or EGF. And this is associated with a moderate improvement in blastocyst development. An alternative approach is to use a biphasic system. Using this we use a high dose of phosphodiesterase inhibitors, and the oocyte is completely blocked at the germinal vesicle stage for several hours, maybe even 24 to 48 hours. So completely arrested at the germinal vesicle stage by the phosphodiesterase inhibitor. That's then washed out and oocyte maturation is allowed to resume. So this is associated with an increase in blastocyst production as well. So there's been a system that's been around for a long time, really, of using very high levels of cyclic AMP to try to mimic what happens in vivo. So using exogenous cyclic AMP, this leads to a very large increase in cyclic AMP, which is followed by a fall. And I should note that the levels of cyclic AMP throughout maturation are still higher than they are under spontaneous maturation. And this is associated with a further improvement in embryo development. Now, to show you some evidence that this works and that in fact the evidence is substantial, I summarised this in a recent review. These are some of the cyclic AMP modulated IVM systems that have been used across the ones that I've just described: biphasic IVM, induced IVM, high levels of cyclic AMP using molecules such as dibutyryl cyclic AMP, invasive adenylate cyclase, or forskolin. So you can see here that in general, across many species, there's really quite substantial improvements in the IVM efficiency simply by managing cyclic AMP during the oocyte maturation period. And I'll point to this study here by Funahashi, which was what, 20 years ago or something, where he used dibutyryl cyclic AMP. And this has really become a very, very successful approach that has been around for a long time. So we use a similar approach to this using forskolin and IBMX in a pre-IVM phase. This is the so-called SPOM system. And you can see here that in the mouse, from this publication here, when we used high levels of cyclic AMP in pre-IVM followed by IVM, you can see that compared to conventional IVF or standard IVM, we have a really quite really nice improvement in fetal yield here. So this is day 18 of pregnancy. So there are many publications here that have investigated this, and to summarise a very large body of work, we know that ovulation is associated with high cyclic AMP, as is induced IVM systems, that is with high cyclic AMP. We get an improvement in embryo development and an improvement in fetal viability associated with some of these mechanisms. So firstly, a preservation of gap junction communication, and a change in oocyte metabolism, so an increase in glycolysis and an increase in oxidative phosphorylation, increase in glutathione, activation of the EGF-like peptide cascade. Very importantly, as Marc-Andre showed before, we have an ordered cessation of messenger RNA synthesis inside the oocyte, so the oocyte goes from being transcriptionally active to silent during the arrested phase. We have delayed germinal vesicle breakdown, and quite importantly, we have a decrease in meiotic synchrony. So there's a substantial amount of knowledge and evidence here of what we're doing to the oocyte. I'm just going to show you simply two examples from some of these studies. So here, for example, these are cattle oocytes, cow oocytes. So when we take the oocyte out of the follicle and put it in vitro, we immediately lose gap junction communication with the cumulus cells. So this is disastrous for the oocyte. The oocyte needs its cumulus cells, and if the oocyte is not in the correct stage, ready for meiotic resumption, loss of communication with the cumulus cells is disastrous. So without any phosphodiesterase inhibitor, we lose communication with the cumulus cells. Using high levels of cyclic AMP, we maintain communication between the oocyte and cumulus cells. So this is over 6 hours here of pre-IVM with high levels of cyclic AMP. This is associated with accumulation of glutathione in the oocyte. So what we can see here, here is the control levels of glutathione in the oocyte. If we block gap junction communication, we have a reduction in glutathione levels. Increasing cyclic AMP, we have an increase in glutathione, and that increase in glutathione is dependent on the presence of communication with the cumulus cells. So cumulus cell communication to the oocyte is vitally important. And you can see that there's a time-dependent effect here. So this is the effect of the duration of pre-IVM with high cyclic AMP, 2 hours, 4 hours, 6 hours. You can see that we have this continual accumulation of glutathione in the oocyte. Glutathione is such an important molecule for oocyte quality. We've been interested in the metabolism of the oocyte for some time. So when we treat the oocyte with the COC with forskolin and IBMX, of course we are stimulating adenylate cyclase, inhibiting the phosphodiesterase. So you would expect, by producing very high levels of cyclic AMP in the cumulus oocyte complex, that we would have ATP depletion in the oocyte, and that's indeed what we have. So you can see here, this is a recent publication. We developed a novel mass spec method to be able to measure all the adenosine nucleotides in the oocyte, and you can see here that with pre-IVM in blue, we have a depletion in ATP in the oocyte. And you would expect, of course, there to be an accumulation of AMP. We weren't able to measure AMP inside the oocyte using this method, but we were able to measure ATP, and you can see here again that with pre-IVM with high levels of cyclic AMP, we have an accumulation of ADP inside the oocyte. So this is where cyclic AMP is being degraded or hydrolyzed to AMP, and then it's being recycled back to ATP through the adenosine salvage pathway. So therefore we have a change in the ATP to ADP ratio inside the oocyte, and when we have a change in the ATP to ADP or ATP to AMP ratio, this means we're going to have activation of AMPK. AMPK is one of the really fundamental energy sensors in cellular systems, and so what we did was we measured AMPK. So this is pre-IVM with high levels of cyclic AMP. You can see, curiously, that at 2 hours of IVM, this is in the mouse, we see no activation of AMPK, but by the end of maturation, you can see we have very substantial activation of AMPK in both in terms of total AMPK and phosphorylated AMPK. I emphasize this is within the oocyte. If we look at the intact cumulus oocyte complex, very strangely, we have an increase in ATP production in the whole COC when we have a pre-IVM with high cyclic AMP. So even though we're driving an increase in cyclic AMP production, we're also producing an increase in ATP in the cumulus cells. So this suggests that we've got some very major metabolic changes occurring in the cumulus cells with this system. Consequently, we have no major change in AMPK activity in the whole COC. So the consequence of this is that when we have high cyclic AMP levels in the pre-IVM system, in the cumulus cells we have an increase in ATP, whereas in the oocyte we have a decrease in ATP and associated increase in activation of AMPK. Together, this is associated with an improvement in oocyte competence. So it's fantastic to have all these great advances in animal studies, and there's really been substantial progress made over the years in animal studies. What about the challenges of getting this into the clinic? And this has been really an area that I've been working on for many years, and I think apart from the biological challenges, which is what I've been talking about here, the actual practical challenges of getting that into the human IVM clinic are absolutely enormous. And this is sort of obvious, but it needs to be stated. Firstly, human oocytes are scarce and precious, so it's very difficult for us to do human oocyte research, and we don't want to be using the discarded oocytes from IVF. We don't want to be using the leftover stuff, okay? We need to be using good quality oocytes, and that's very hard. Very importantly, we have very poor measures of oocyte quality. So this means that if we're going to make any changes to an IVM system, we need to produce human embryos for research purposes. And of course, that in most cases, we can't do that. So there's a very, very substantial barrier right there. The third barrier is that obviously the IVF status quo works. So there's really quite substantial resistance from the ART sector now to make change. So I think to take these very substantial advances in the animal sciences into the human ART sector is going to require some very, very brave efforts. So one group I think that is doing this as well as anyone is the group at VUB in Brussels, headed by Johan Smits. I've been working with this group for 10 to 15 years or something. And they use some very important models. Firstly, they take PCOS patients, and they encourage those patients to donate half their oocytes for research. So all the oocytes from one ovary are donated for research. Secondly, very uniquely, they have PCOS volunteers. So these are young women, typically nursing and medical students, that are paid to donate their oocytes for research. And this enables us to do some really good quality research, although still limited, using these good quality oocytes. Secondly, in Belgium, and there's not many countries in the world where we can do this, we can generate embryos for research purposes. So using this type of approach, in the system in Brussels, 3 days of HMG, no HCG priming, oocyte pickup 42 hours after the last HMG injection. The follicle size is very small, so less than 5 millimeters. Typically 5 millimeters, but so less than 4 up to 10 millimeters. So using the type of IVM systems that I've been talking about here, we've done a study where we've used pre-IVM system using forskolin and IBMX compared to together with Coumadin. And I haven't spoken about Coumadin, but Coumadin is a heterodimer of GDF9 and BMP15. And you can see here that when we add it to the IVM phase and then look at subsequent development, we have no change in M2. We have a slight but non-significant improvement in fertilization rate. We have a significant improvement in day 3 embryo quality from M2 oocytes. We also have so that's rate. This is quality of day 3 embryos. And using these very low numbers we have so far, we have an improvement in blastocyst production, but not significant at this stage. I'm nearly finished, so just a final slide. I'd like to point to a very important development, I think, which is along these lines. It's again from VUB Brussels, using this type of oocyte capacitation system in vitro. So instead of using a phosphodiesterase, a pharmacological phosphodiesterase inhibitor, the group in Brussels have used C-type natriuretic peptide. So this is the molecule that stimulates cyclic GMP, so here it is here, stimulates cyclic GMP production, so it inhibits the oocyte phosphodiesterase through cyclic GMP. So this is a biphasic system where we completely block meiosis for 24 hours, so using CNP. Estradiol is quite important, a very low dose of FSH. Then those oocytes are washed out. The oocytes then undergo germinal vesicle breakdown, and oocyte maturation is induced with amphiregulin. The dose of FSH that's used here is very low, so oocyte maturation is induced by amphiregulin, which is the natural meiotic inducer in vivo, and then the oocytes are fertilized. So here are the results. It's published just recently in Human Reproduction. So this is the standard IVM system that's used clinically in Brussels compared to C-type natriuretic peptide system. Quite importantly, you see an improvement in the metaphase 2 rate. You see an improvement in 2 cells, development to day 3, and a doubling in blastocyst production. So this is really very encouraging. It's very, very difficult research to do, but you can see here by using some fundamental principles based on good science, you can see that we have the capacity to try to improve human IVM. So my last slide, how do you make a good quality oocyte for ART? Well, you can stimulate the patient with a lot of FSH and a lot of HCG, and this system works. There are problems associated with this, as we know. It's also expensive. It costs either the patients or medical providers a lot of money. So the alternative system is you don't stimulate the patient with FSH or HCG, but rather you take the oocyte out and you treat the oocyte in vitro. Okay, so you don't stimulate the patient with extensive FSH. You would treat the oocyte with meiotic inhibitors such as cyclic AMP or CNP, and very importantly, you need to support the cumulus cell development with molecules such as Coumadin, EGF-like peptides, and FSH. So I'd just like to thank, finally, I'm sorry if I've gone a bit over time, not too bad, I think, my current team at the University of New South Wales in Sydney, my old group at University of Adelaide, the fantastic collaboration I've had for many years with Johan Smits and Michelle DeVos, really fantastic work here. Some of my old friends in Canada, the new team in China, and also funding from NHMRC in particular, and also from Cook Medical. Thanks.